Evening, everyone. I welcome you all in today's demonstration on single molecule imaging under high-low illumination mode and its tracking analysis. So previously you have learned about fluorescence microscopy, confocal microscopy, wide field microscopy. And in today's session, you are going to learn how you can image and track single molecule and its analysis. So for this imaging, we are going to use this Leica DMI-8 system with Prime 95B camera. And along with this, we will use laser systems and turf controller. So here we have kept this sample. Rashmi, can you bring here? Yes. So this one is bacterial sample, and we have tagged DNA polymerase gamma protein um, with halotype. Okay. So here Nitesh will show you how you can record movies and how you can identify signal at this. Okay. How you can record movies and how you can visualize the single molecule of protein. Okay, so Nitish, can you start?
Yes. So today we are going to learn about single molecule imaging and using high-low microscopy. So for that, we have taken a bacterial sample where we have tagged a protein, DNA polymerase gamma with halotype. So for that, what we need to do first, we have to take the sample. Okay. So basically, we are using 100x start objective to do the single molecule imaging. Okay, so let me so okay, so we are going to learn today how to do single molecule imaging and tracking that we used to do in our lab. So for single molecule imaging, we need two things. First of all, we need to tag a protein with halotag level because halotag is having high intensity, so we can capture at single molecule level. Second, we use the illumination. Now, if you have learned that in normal epifluorescence mode, we illuminate the entire samples. However, in high-low mode, it is highly inclined laminated optical slit microscopy that I show you in some time, like how we'll set and adjust for the high-low illumination to image our sample. So now we'll uh, go for that.
So we have turned off lights, and here you can see
So
this software.
So this is so coming to this, now you can see, if you see the graphical interface will look like this. Now this is the software interface. Now if you see here, there are certain parameters that you need to set to perform single molecule image. Now for that, what is the exposure time? Okay, so we used to capture images at different imaging regimes. For example, suppose you want to capture diffusion parameters and diffusion dynamics, then you have to capture first imaging. You have to go for less exposure time. So we capture first images, keeping the exposure time very less. However, to calculate or estimate residence time, we use higher exposure time, that is 50 millisecond, and it's a long interval maybe. So we we capture 200 millisecond interval movie to estimate the residence time and other parameters. So now if you see, we have to do the alignment for the laser before starting the single molecule imaging. So what that means, that what laser alignment means, you have to align the laser through the objectives. And now if you see here, if you can see this torque configuration mode, now here you can see there is a long line having partially green and partially this black mode. So now if you see, if you can see here, so this is like here I'll show you. Okay, so now I just will focus on a cell. Okay, so
Right. It says that.
okay, so we'll go for a larger field region so that we can.
Right. Yeah. Tell them which bacteria it is.
Okay, so this is
Bacillus subtilis.
Bacillus subtilis, and here we are going to.
This
Now we are going to capture the single molecule movies at 200 milliseconds. So now, yes. So what I want to say is to capture single molecule movies. Now, if you just try to focus on this panel. Okay? So just try to focus on this panel. If you can see here, I'll just close. So now you can see here there is a red line, and these fractions is basically the TURF mode. Now, TURF is total internal reflection fluorescence microscopy, where the light passes at a critical angle and the evanescent waves that hit the platform will illuminate your sample. Now, in this mode of fluorescence, we can only capture a sample till 200 nanometers. So, in order to capture the internal structure, for example, some nuclear fluorescence or cytoplasmic fluorescence, we have to use this halo mode is more efficient as compared to the turf illumination. Now, if I'll sit like this, so this is the epifluorescence mode. So more I'll go towards uh, right side. So this is complete epifluorescence where the entire sample is getting illuminated. However, in the green zone, it's a turn. Now to set halo illuminations, we have to set in between this. Now you can see here, there is an angle. Okay, so basically this is the halo angle. So I'll set this parameter just closely to the halo angle. And now if you try to focus on the entire screen, so just you can see while I am capturing the samples, for example, if I go to the epifluorescence, now you can see the signal to noise ratio is less. Okay. However, if I'll go to turf, also you will not be able to capture. But if you'll go to the halo mode, this is the most appropriate one where you can capture the signal to noise ratio, as you can see here. Okay, so here, so the signal to noise ratio will increase in halo illumination mode as compared to the uh, epifluorescence illumination, the white field illumination. Now, this imaging we capture for different time regimes to capture different biophysical parameters. For example, for 200 millisecond, we capture a 200 millisecond interval to capture the residence time, and we capture continuous images to calculate the diffusion parameters. Now, as you can see here, now this is the image has been captured, and now you can see the single molecules, the blinking of these molecules here and there in this plane. I'll just try to show you some good images also. Okay, now here you can see. Okay, so you just try to focus on these cells. Can you see the particle moving here and there, right? So basically now we are going to form trajectories out of these movements. We are going to capture this, dynamic informations of these single molecules of these particular proteins. So basically we're going to track these molecules, track these trajectories. So we are going to connect this frame to frame of these molecules to form the trajectories, and we are going to track their, calculate different biophysical parameters of this. So to understand that, now I'll go to make you understand through the softwares that we use to do the tracking for single molecule movies. Okay, so now I'll share my screen to make you understand more about how we track single molecules using MATLAB-based software, that is track record. So just share my screen for that. Okay. So hopefully my screen is visible to all of you. Yeah, it is, right? So now here is the movies that we captured. Now if you can see, I'll just play the movies for the 200 frames. So this is a movie that we have captured to understand the residence time analysis. Now if you see this movie, okay, so you can see the interaction of these molecules, right? The movement of these molecules, I'll just select the brightness and I'll just adjust the brightness and contrast so that you can properly visualize the molecule. Okay. So now our purpose is to connect these molecules as to form the trajectories to understand the residence time or different biophysical parameters. Basically, we use this platform or this um, track record. This is the MATLAB-based software called track record to understand to estimate the residence time from these tracks. Okay, so I'll just so what we do first we upload the images. Okay, so this is a time-lapse movie. Now what I will do, I'll filter these molecules using bandfast filter. So I'll just filter the molecules. Now here you can see the filtering is going on. Now you can see the filtering is done. So what we'll do, we'll add ROIs. So basically what is a ROI means? Suppose I want to capture my molecules or I want to track my molecules at a particular region of interest. For example, here if you see the plane, this is a yeast sample having cells at different cell cycle stages. Like this single dot represents cells at prophase, these are cells at metaphase, these are cells at anaphase with the spindle structure. Okay. Now I want to track my protein. So for example, at the spindle. Okay. So we are going to make region of interest around these spindles to capture the trajectories that are interacting or that are to understand the dynamics at this particular location. So here you can see I have made ROIs at the spindle. So I'll add another one. If you can see here, here is one more anaphase cells, right? So what we'll do, we'll make region of interest around this particular, wherever we want to make our region of interest, wherever we want to track our molecule. Then I'll go for find particles. Okay. So we have added the ROIs. So I have to add one more ROIs. Okay. So we have made the ROIs. Now we'll set the threshold. For example, we'll set first some random values to understand like what may be the best threshold to detect our single molecules. Now I'll click on find particles. Okay. So it will find the particle. Now, if you can see the finding particle is done. Now we basically keep certain criteria to define whether a molecule is bound or not. Now, if you can see this, we are asking the software to consider only those molecules that are residing within a maximum of 6 pixel, and the shortest track represents that what is the shortest track that we are expecting. There are sometimes what happens, the software will connect one or two molecules as a track, and that we don't want because that will impact our results. So we want only those. Yes. So we want only those tracks. Okay, so we want only those tracks, those are the actual tracks basically. So, and this gap to close is sometime, since we are imaging at a 2D plane, sometime the molecules move to some other plane. You are not able to visualize the molecule while capturing the single molecules. So sometimes the molecule again will come back after 2, 3, 4 frames. So we want our software to also consider those tracks that are those molecules that are not for few frames, but again coming back to your plane of which you are visualizing. Okay, and then you want your software to track. So you are basically keeping some criteria to consider bound or unbound molecules. So basically we perform tracking, then we'll go to process track. Okay, now what we did, see if you can see, we process the tracks, and now what our software did, just that's really interesting. You want to see, just I'll show you. So if you can see this, now this is the one that our software generate the tracks. Okay, so now if you see, I'll zoom this. Okay, can you see these tracks? So basically, yes. So basically, the software connects these frames using nearest neighbor algorithm. So basically, it connects individual the same molecule at different frames to form a trajectory. So basically, our end product is this trajectories, and from there, what we can conclude, like for how long a molecule is staying at a particular location. Okay, so this is how we track our single molecules using single molecule imaging platform. So there are so many softwares that can be used for estimating different biophysical parameters. So this is one of that we use MATLAB based track record to estimate residence time. Okay, so now you have to analyze individual time-lapse movies to capture these parameters. And once you are done with that, like for example, I showed you how to analyze the single movies, right? Now you analyze these particular movies to generate those tracks. Now, once you make the tracks, now we keep certain criteria. Suppose for my model system, we have a standard values. For example, I will track for 1,000 tracks. So we generate the data for 1,000 tracks. And from there, we'll estimate like what percentage of molecules are bound molecules, and from that what percentage of trajectories are residing for how long. Basically that will be our end product. Okay, so from there we can be able to estimate the residence time of a particular molecule that is responsible for certain biological functions and so on, that we can conclude out of that. Any questions? Mhm. So no doubts. now I'll just show you like for how we generate some end products from our old data. So now we have to go to further analysis and merge data and analyze residence time. So basically you analyze individual movies, and after you once you are ready with all your movies to 4,000 tracks, then what do you have to do? You have to go for the like then you have to analyze, you have to merge all these movies to generate some data. Okay, so the end products will be like, so we have to go for, suppose now I'll show some already done experiments data. So this is what you will get at the end of this. So basically at every stage you have to set certain parameters to differentiate between your bound molecules and unbound molecules. Okay. Now for that, we have to take some as a some control. For example, you want to categorize histone bound molecules. So for that, a chromatin bound to you know you have to always take some gold standard stable bound molecules. For example, to categorize the chromatin bound molecules, we track histones. For example, to categorize kinetochore bound protein molecules, we have to track certain kinetochore proteins, stable proteins, for example, NDC10. Now here, from there, you have to define certain parameters. For example, here you can see maximum jump. Now this maximum jump is from frame to frame displacement of almost 90% of the stable bound molecules. So these parameters are for a stable bound molecule. So I'll put some histone bound, matlab stable bound parameters for our histone molecules to track these to get our end results. So here I'll put the standard values for that, and I'll show you that how the end product looks like. Okay, so these are few parameters that we put, like we captured 200 millisecond movie, so we kept 0.2 as frame to frame time. The minimum length of bound tracks, that is basically the number of frames till which your molecule should not exceed the armin or this maximum jump. And this end-to-end displacement is the displacement or displacement between two consecutive points within the n min number of frames. Okay, so now I'm going to put those values and we'll try to see what we are getting. So it will take some time. Okay, so this is here. If I'll show you this data, now you see this. So basically, at the end, you are going to get this kind of plot. Now, if you see this here, this is the for the this is the time for, and this is in y-axis. You can see the count. So basically, the 0.7%, 0.7, this is the probability basically. So this 0.7% of the molecules having a residence time of this. Okay. So basically, this is a cumulative curve. Okay. Now, if you see this data fits properly to the red line, that suggests our two component fitting. So what we are expecting now? Now we can expect there should be two different population or different fractions or different kind of bound particles should be there. So while I'll generate the pie chart, now if you can see here you have a yellow fractions that shows that this 30% of the molecule or 31.9% of the molecules having a residence time of 9.78 second, and the 36.1% of the molecule having green fraction that represents the transient bound molecule, or these are the molecule interacting transiently having a residence time of 1.2 seconds. And this 31.9% of the molecules are the completely diffusible molecules. So basically, this kind of parameters that you can estimate using this kind of using this software, that is the residence time and all. So finally, you can calculate these parameters for different conditions and different uh, for your different experiments, and you can compare those to understand how this dynamicity in context of residence time is differentiating or different from sample to sample or from conditions to condition, and decode certain biological output or biological functions. Okay. Everything is clear now? Any questions? So basically, you can inquire for anything about single molecule imaging and tracking. You can also visit our lab websites to study or understand more about these techniques. And this is one of the robust and latest techniques that people are using across the globe to understand the dynamic differences between the molecules. Because if you see the average estimation based techniques like Western and all will lose the dynamic information. Okay, so to capture such fast dynamics, we use this single molecule tracking approach to study the dynamic interactions and study the change in dynamicity in context of dynamic differences that we cannot capture using the average based techniques. So you can get in touch in case you need more information about single molecule imaging and tracking,
So, uh,
and you can also write in mail to understand more about this. Thank you everyone for the session. Yeah. Now, कैसे करते हैं? Message में.
हाँ सर,
Thank you, sir.
it's a blue line. As you reflect to the
But चला गया ना ये. Okay. Okay, I'd like to discuss about blue line, right? Okay. So basically here, if you can see this plot, I'll just share my screen again just to uh, tell that. Okay, so this is the plot. Okay, so now see this. Now, yeah. So here you can see two blue line, right? So this two blue line represents the fitting for the individual components. Okay, so this is for one component and this is for one component, and now this red line represents the cumulative fitting of matlab, complete fitting of the final fitting of these two fitting. Okay, so this is the two components. So this green line and red lines are the single and double exponential fit. However, the blue lines are the fittings for the individual components of this uh, to make this single, whether it is fitting to single component properly or double component properly. Okay, is it clear? Okay, anything else? Okay then, thank you so much, Arjun. या करना क्या है अभी?